excited to introduce from the Jolla Institute of Immunology, Dr. Tal Einav.
All right, thank you very much, Andrea, and thank you all for being here. I'm very excited to present at this beautiful theater. So my name's Tal Einav. I am the newest faculty at the Jolla Institute for Immunology, and I have a bit of an unusual background for an immunologist. I was trained as a mathematician and physicist. I switched lanes into biophysics in my PhD, and then one more time in my postdoc to immunology. So the way I think about questions, the way I tackle them, and what I think is big and fun and interesting science is going to be slightly unusual. And so just to give you a sense of what my group is all about, we love data. We're all about computation and sweeping in the data that we've learned already in order to make new discoveries. So the central question of my lab is, how can we use vaccine studies from the past, the decades and decades of hard work that everyone has done around the world, to predict your specific response to future vaccines? And this is a challenging problem because each of us are unique and different and special. And so, for example, if we think about viruses that infect us, we mount these strong antibody responses against them. And our antibodies are these very special systems that can do almost anything. They can bind to any pathogen that infects us, no matter what it is. And so if we think about one of the antibodies in your body, there are about 120 different amino acids that comprise how it binds to foreign substances. And each of these 120 slots has 20 possibilities. So there are 20 times 20 times 20 times 20, or 20 to the 120th possible ways to make one single antibody. And in your body you have hundreds of thousands of different antibodies. So it's this big, complex mix where every one of the pieces can be unique, and this is really what defines you and how well you're protected against the different viruses that will infect you. So this number, 20 to the 120th, is very large. It's in fact much larger than the number of stars in the sky. It's much larger than the number of atoms in the universe. And that means that no matter how many experiments we do, no matter how many variants you test, there are still more antibodies out there that can do new and interesting things. So how can we take the data that we have, how can we take the antibody responses from a few individuals and understand how each of us will behave and react? So that's what my lab does. We study viruses such as influenza, HIV, SARS-CoV-2, and the antibody responses that we mount against them. And if we just think about flu, so the flu vaccine that we get year after year, we mount these complex antibody responses that are highly dynamic. So wherever you are right now this year, as we go year by year and you get your next vaccine and the virus evolves and you get your next vaccine and on, or you get infected every so often, your antibody response changes with each step. And people are interested in understanding this process from both sides. So how, for example, will the virus evolve? If we take 2 steps back and think about January, these are the 2 viruses that we had circulating around the world. And we want to be able to predict what's going to happen in November, because we get our flu vaccines right now, around November, December, January, but we have to say what should go inside the vaccine back in January, back when we had only this information. So we have to predict where is the virus going to go with each additional step. And groups around the world are specialized to predicting what happens with the virus so that we can pick the right one to put into the vaccine. But the tricky bit is that even when we predict correctly, when we think that what's going to happen in November is exactly the virus that we see back then, we still see that when we give the vaccine to many different individuals, the majority of them will mount a very strong response, but there are some that mount a weak response and a few that show little to no response. And so we want to fix that. We want to be able to minimize the number of people in this group and shove everybody upwards so that everybody gets the best possible response that they can. And so the question of my lab is, what happens if we focus on this bottom part of the presentation? If we think about what your antibody response looks like when we give you one vaccine versus another. And that's the prediction problem that we want to make. All right, so here's the goal. Shown in a slightly different way. We're going to give you a vaccine, we're going to measure your response, and we're going to see how do your antibodies inhibit different viruses over time. So right when we give you the vaccine here, we're going to measure your response over and over and over and over again and see how well do you inhibit the virus, the inactivated virus that we put into the vaccine that we want for you to mount a strong response against. So higher is always going to be better on these plots, and we would love to see this big, strong boost in your response that ideally lasts for a long, long time, perhaps years, perhaps your entire life. So a big, strong response that slowly, slowly, slowly goes down as time goes on. And the challenge is that even when we make such a vaccine and it really works well for you, the neighbor sitting right beside you might have a somewhat different response. And even people who seem to look like they start off at the same point with this weak inhibition might mount very different responses. Some of them don't do anything, other ones mount a medium response, some of them mount this strong response. And so this has been the challenge in the field. How can we understand the different responses that each of us elicit to the exact same vaccine? So groups have gone off to measure all kinds of different facets of our immunity. Some of them look at age. Some of them look at prior infection history. Some of them look at your genetics and ask, are there different parts of your genes that tell us who's going to respond strongly and weakly? And a really interesting metric that people have started to use several decades ago is that we measure the responses not just against the vaccine strain, the inactivated virus that we put inside the vaccine, we also tend to measure now against other viral variants. Because if we go back and we think about what the virus is doing, it's mutating all the time. And even though we gave you this one particular variant, there's all these other variants that we know that you have seen in the past, and there's all the potential variants that the virus might be in the future. And so every single study out there is going to try to probe the diversity of viruses. One study is going to look at these 50 viruses. Another will look at those 50. A third will look at its own 50. And there might be a little bit of overlap between each of these studies, but it's not complete. And this has really been the primary barrier to understanding and looping together all the studies that we've had in the past. The fact that everyone measures something slightly different. And so this talk is going to be about that specific challenge and how we're flipping it on its head and not looking at this as a detriment that keeps us from combining data sets, but rather as an asset where we can now predict much more than we could have done before, including how your specific response will be this year to our vaccine. All right, so I'm going to split this talk into 3 vignettes. The first one will talk about how we can predict new virus behavior using machine learning, not just to predict how a virus will behave, but even to quantify our uncertainty in these predictions. This is state-of-the-art methods that we're then going to apply to different data sets out there, and I'll show you what happens when we make these predictions. How good do they behave in the wild? And finally, I'll go back to your specific response and mention what happens when we give people different vaccines. How will they respond, and how can we predict that ahead of time? Okay, so starting with the methods. So this is now going to be the theory part of the talk. Brace yourself, no equations, but a lot of really interesting science. So I'm going to rephrase the problem one more time from a visual perspective, where I want to show you what do the data sets look like that we play around with all the time. And these are just little snippets of them, but imagine that you get one data set that is going to have hundreds of different individuals. I'm showing 5 here, measured against 50 different viruses. I'm showing 20. And each of these squares represents how well does this person's antibody response inhibit this specific variant of influenza or flu. So for one person, we'll measure how well do they inhibit virus 1, 2, 3, 4, on and on for every single virus that we've measured in this study. And we do that for every single person. And then there's a second data set that measures its own hundreds of individuals against all these different viral variants. And so each one of these will be some number that I'm just hiding from you for convenience at the moment. But the key that I want to really point out with these colors is that the blue viruses in the middle were measured in both studies, whereas the grays were only measured upstairs in dataset 1, the greens were only measured down in dataset 2. And so traditionally, if we would just think about 2 years ago, if you wanted to combine the power of both data sets, you would focus on the blue viruses. You have no choice. Those are the variants that were measured both times. And so you can say everybody who was measured here and everyone who was measured there, what do their responses to these 10 blue variants look like? But you can't use any of the information from the grays down here, because we don't have them, nor the greens from up here. And if you think about data sets 3 and 4 and 5, they will have their own variants that they measured. And if you only look for the overlap, for what was measured in every study, you're going to quickly wheedle down and down and down and down until you have almost no data left. So that was traditional state of the art, where we said, well, you either analyze this data set by itself, or if you want to combine studies, then you have to look at a smaller chunk of data and then make do with the best that you can. So we came in and we said, well, that seems kind of strange. Why don't we try to predict how the green viruses would have behaved up here in dataset 1, how the grays would have behaved in dataset 2. Because if we can do that, all of a sudden we gain the power of both studies. The first one up here would enable us to take anybody down here and predict how would they have behaved against the grays, the blues, and the greens all together. And the same thing up here. Now we can predict the grays, the blues, and the greens. And then when you add a data set 3, 4, 5, all of a sudden you just gain more and more and more with every additional data set that you add. So this is the problem that I want to discuss with you here. How do we predict these dark red values in the corners? But as a step towards that goal, let's focus on the blue viruses just for a second. Let's withhold one of these viruses as if nobody measured it in dataset 1. And we can predict those values, and we can see how well we did at the prediction challenge. And then we'll do this again and again for every variant in every single study to see how robust our prediction algorithm is. So this is the name of the game. We want to predict this dark blue virus in dataset 2. And the challenge is that we don't know what these values are. We can't use them, but we know how this virus behaves in dataset 1. And we know how all 9 of the other blue variants behave in both datasets. So if we can find a relationship between these 9 variants and our variant of interest in dataset 1, where we have all the information, we can copy and paste that into dataset 2 and predict the variant that we want from the data that we have. And this is where machine learning comes in, and it's a very powerful tool. It lets us go to dataset 1 and grab some of these variants, grab some of the antibody responses, and form a training set, meaning that we train a model that takes as input how any person out there, you, me, your neighbor, would inhibit these 5 specific variants of flu. Think about this as flu from last year, the year before that, the year before that, and on and on and on. And it predicts how will you inhibit this year's strain. So it's exactly what we want to predict. And if we do this, we train our what's called a decision tree algorithm. I won't go into the technical details other than to say it's a fast and highly interpretable form of machine learning that takes as input how anybody would respond to these 5 variants and outputs how they would inhibit this final variant of interest. And so we train this model, and then we want to see how well does it do. So we're going to take all the antibody responses that we haven't used, all this unused data, and that forms what's called our validation set, where we hit the model on this new data it hasn't seen before, and we ask how well did it do? What was the training error from the training set? And we'll do this process many times because we don't know which of these viruses are the best, the most predictive ones for our variant of interest. But we'll do this hundreds of thousands of different times. The computers chug away, and then they tell us what are the best, most predictive variants for our specific virus of interest. And that's the model that we'll ultimately use in dataset 2, using the information from these variants that we have down here in order to predict the variant that we want out here. And so that was the goal, and that was the moment of victory. And if you had asked me 2 years ago, this would have been state of the art for how we can predict these values that we want. But the problem with this method is that it's missing one key feature that we need to make it practical. And that is that we need to be able to estimate not just the values, not just how well do people inhibit this particular virus, but also we need to understand what is the actual error, the accuracy of our predictions. So if we could just for a second peek behind the curtain, if we can see what these values were, then we can plot the measurements against our predictions, and we can tell you we're one-fold off or two-fold off or however off we are from the actual data. But we can't. We don't have this information in order to directly assess our model. So the key question of this analysis is how can we estimate this actual error? Given the fact that we made a machine learning algorithm in dataset 1, we even tested it here in dataset 1, and we saw how well it works. So all of this is done in dataset 1, and then we apply the model to dataset 2. And we need to understand how well will this model work in dataset 2. Will it be fantastic or will it be tragic? Because sometimes these two data sets can be very different from one another. Perhaps we're using data from animals to predict the response in humans. Or perhaps we're using data from children to predict the response in adults. Or perhaps we're using data from the US to predict responses in Australia. Will that matter or will it not? Will they have the same responses or will they look very different? There are times when you can expect that a model might perform fantastically in your dataset 1, but as soon as you try to port it over into a very new setting, it will start to behave very poorly. So we have to know what is the error of our model in dataset 2. Just to take an analogy here that might be more familiar, if you all know about ChatGPT and how it can now answer questions for you, you can ask it a question like, tell me about sights to see in Rome, and it'll give you 10 fantastic places to go to see. But if you ask it a specific question like, cite the 10 most influential papers that tell you which sites to visit in Rome, it can absolutely make stuff up, and you won't know it. It doesn't come up and say, well, I don't know about this specific answer, but here's my best guess. It just gives it to you via the very straight expression, doesn't give you any hint whether it knows or it doesn't know. So we're trying to give you that hint at this point. We're trying to say, here's what we think will happen when you use your data set you use your model from dataset 1 into dataset 2. Either we might be super confident if the error is very low, or if the error is very high, then we'll tell you, well, we just don't know, but here's our best guess. So how do we solve this problem? What we said is, let's take our model that we're training from dataset 1 to dataset 2, and for the moment, we'll just ignore the virus that we're trying to predict. We'll focus on the other 9 variants that we have, and we'll try the same algorithm once again. We'll take one of these variants, and we'll predict it from dataset 1 to dataset 2, except this time, we actually have the measurements right here, which means that after we do our predictions, we can compare them to the measurements, and we can see what is not only the training error in dataset 1, but what's the actual error in dataset 2. And if we do this for every single one of our variants, going from dataset 1 to dataset 2, then we calculate what we're calling as the general transferability of information between these two datasets, which just means how does the training error in dataset 1 correspond to the training error in dataset 2. So if the two datasets are the same, if they were both measured in the US vaccine studies on adults year after year, then we expect that exactly what you see in training error up here will be the actual error down here. You can use the same model no matter on which dataset you go. But when the two datasets are very different, when dataset 1 is very clean data and dataset 2 is very noisy, we will understand that these two datasets have a different relationship, where a small training error can lead to a large actual error. And so once we've quantified this relationship, we can go back to our virus of interest, calculate its training error in dataset 1, what we always have access to, plug it into this relationship, and estimate the actual error in dataset 2. And so that for us was true victory here. It meant that we can go back and predict for these cases where we have somewhat overlapping datasets, we can predict for these green and these gray viruses, how would they behave? And not just get their values, but even estimate the error or the uncertainty of the predictions, so we can tell you which of these variants can we exactly predict, and which of them are just very hard to predict, so we have high uncertainty in them. Okay, theory part over. So that is what we developed in terms of this machine learning algorithm. Let me show you how it works on these data sets and how good the predictions turn out to be. So we grabbed some large-scale vaccine studies done in humans, as well as infection studies where you track people over time. You see how often they get infected. And we threw in this ferret antibody data set just for kicks to see how different are humans and ferrets. And we wanted to know, how do these data sets talk to one another? How do they relate? Could we take one of these studies, completely wipe off one of these rows as if they hadn't measured this virus and predicted back, and then do that for every single one of these variants that they measured in this data set, and then do that for every one of the variants measured in any of these data sets. So altogether, this is going to give us 200,000 different measurements that we can predict. This is 10 years of experiments. If just a person in the lab is not sleeping, not eating, and they're just pipetting up and down, that's how long it would take them to do this many experiments. We're going to do it in a few minutes using our computations and leveraging the fact that all of these studies measured viruses that were somewhat overlapping. Okay, so here's what those 200,000 results look like in one gigantic plot, where I'm going to, just to reiterate, we don't just give you predictions, we also give you an uncertainty estimate. So if a measurement here is 10, signifying a very weak antibody response, then we want to predict something between 5 and 20 most of the time, which is going to correspond to twofold error. This is what we predict is going to happen across the board for our 200,000 predictions. So a measurement that's weak, like 10, is going to be between 5 and 20. A measurement that's strong, like 160, is going to be somewhere between 80 and 320. That's what, before even showing you any data, that's what we predict should happen. That's what we estimate should be our uncertainty. And now I'm going to show you from each of these studies what happens when we pull out the weakest measurements, the ones where the measurement, the experiment said a value of 5, almost no measurable antibody response against this virus. And here's what the predictions look like. So the experiments were 5, 5, 5, 5, 5, and the predictions, as you can see here, were mostly 5 down here. Almost all of them were 10 and below, which is this twofold range where we thought they should be. And then there are the few cases where we overpredicted and we didn't do as good of a job. But as I show you larger and larger measurements, you can see that the majority of points fall on this diagonal band right where we want for them to be. And a careful estimation shows that the predictions versus the measurements have twofold error, which is exactly what we said it should have been. So we were very excited about this because it meant that our ability to estimate how good our predictions will be is spot on with what they actually are. So what this means for the field is that when somebody collects a new antibody virus data set, the first question that I want for them to ask is, which of the viruses that we've measured here were previously measured in any other study that happened in the past 3 decades? Because the rule of thumb is that if you've measured at least 5 of these viruses, then we can predict how all of them would look like in your data set. So you get the estimation for each of these viruses, not just their values, but also the uncertainty, which means that we can tell you some of these are going to be poorly predicted, but a few of them will be very nicely predicted with low uncertainty. And so you can take those specific variants, and they form this expanded virus panel that you get for free. We just give you these values because somebody somewhere out there has already measured how these viruses look like and behave. And so we use the overlap between your data set and the previous data set to estimate what these values will look like. So here's what happens when we applied it to our data sets. Here's study number 6, which is my favorite study of them all. You can see here they only measured 6 viruses, but they did it against 25,000 different sera, which means antibody responses from different people. So I'm showing you the data on the left. It basically goes on forever. And if we can add a single virus to this data set, then we would save 25,000 new experiments that somebody has to do. That's a lot of work. And we're not just going to add 1 virus to this data set, we're going to add 75 using all of the other studies that we have. So here are the viruses as much as we can fit on the x-axis. On the y is how accurately can we predict their values. And the results look like this, where 1-fold represents perfect predictions. We cannot do any better than 1-fold. This means that the predictions should be as exact as the measurements there are themselves. 2-fold is the accuracy of the experiments themselves. So experiments have noise. If you redo them multiple times, you'll see in general the answer will hop up by 2-fold or down by 2-fold. So each of these viruses that you see down here that have 2-fold error, we can predict them computationally as well as the experiments can measure them experimentally, which is a total win on our part. And if you allow me just slightly larger error at 4-fold, then we can predict almost all of these different variants that we have up here, except for these 4 troublemakers that we see have larger error. And so we will ignore those in subsequent analyses and say, these are the ones that we can predict with confidence, let's use those. And so my favorite way to plot these results is as a function of the error that we can tolerate. How many new values do we get for these data sets? And the answer in this case ends up being that we get 2 million new values with less than or equal to 4-fold error, which is an astonishing number because the data set itself had less than 200,000 measurements. So what we're doing here is increasing the size of the data by more than 10x, simply by letting the data sets talk to one another rather than just analyzing each of them individually. So I really think of this as an idea worth fighting for. It's a way to make every experiment we do in the lab worth its weight in gold of 10 experiments or more by letting them connect to one another. So just to summarize, we can take these antibody-virus datasets that we have and not only predict new values, but even be able to help design future experiments that are optimized to, if you want to predict these 100 specific variants, here are the 10 that you should measure in order to realistically be able to predict how your full 100 panel would behave. All right, so this is the exciting proof of concept study. And now I just want to shift gears very quickly and show you how does this relate back to you and your specific antibody response, and what are we doing in the lab right now with these types of measurements that we have. So going back to the beginning, each of us has an antibody response that protects us from viruses that we encounter in our lives. And we can get a sense of what that antibody response looks like by taking a sample of your blood, because your antibodies are contained in your blood. And then we can measure how well does your specific antibody response inhibit all of these different strains of influenza. Again, thinking about this as one virus from each year from the past 50 years, and we make these landscapes where larger values means better inhibition. Smaller values means that you're at risk from this specific strain of influenza, but larger values means that you are well protected against this one. And so what we want is that when we vaccinate you, we want for this curve to increase up for as much as possible and as long as possible. And at this point, we have done this process, vaccinating people and seeing what their response looks like for decades. We've done it in the young and the old, across continents, in humans, mice, ferrets, guinea pigs. And the key question that we still don't understand is, how can we predict who is going to exhibit this strong response? Because sometimes when we do the same process, we'll vaccinate someone, but we'll see that their pre-vaccination response and their post-vaccination curves look almost the same, which means that the vaccine wasn't very effective in this case. Almost nothing happened where we really would have liked to see something up here, this big, huge curve of a response. So what happened in this person? What features of this landscape result in them having this weak inhibition? And in particular, if somebody does exhibit a response to this specific vaccine, what we would really like to do is not just predict, aha, the green vaccine would work for this person, but to tackle the more ambitious question, what would have happened if we would have given them a different vaccine with a different inactivated virus? Because that one could have reacted in a completely different way. So there are currently no models that are able to predict how the green versus the orange vaccines would behave head-to-head in the same person. And so that's the challenge that we're tackling right now. Because we're taking these antibody-virus data sets where people measure their pre- and post-vaccination responses of many different individuals in many different settings. There'll be different parts of the world, different age groups, different everything that you can imagine. And if we can learn how does the antibody response change in one of these groups, can we now predict what happens in all the other settings? And the reason why this question simply could not have been tackled 2 years ago is because the actual data sets look like this, where these dark brown squares represent not measured viruses. Because remember, going to the very beginning, every single data set is going to measure their own unique set of variants. And there won't always be perfect overlap between the viruses that one study measures and another. So even if we made a model in this top vaccine study that uses this blue, this green, and this gold virus to predict the antibody response, we couldn't use that specific model in any of these other studies. We're missing some of these viruses and how they would behave in these studies. And so the first step, the enabling step here, is to use the methodology I just showed you that takes the data that we have right here, shown in gray, and expands each of these data sets, predicting how every virus would have looked like in every data set. And now, for the first time, we can take this nice homogenized, expanded data and take our models and learn from one of these settings and predict in the other ones, how do people's vaccine responses look like across different settings? So that's what we're doing. We're taking these huge data sets that we have, and we're creating these universal algorithms that let us predict how any single person out there would respond to a particular vaccine. And we're doing it not just for one vaccine, but for every vaccine that we have and every vaccine that we have had in the past decades, in order to predict how any potential vaccine that we make would look like for any individual. Today, I really talked about a specific vaccine and how it looks like at one snapshot. We're absolutely thinking about what does the response look like over time, because we would love to give you a vaccine that will protect you for the rest of your life. We're also asking if we can predict for each person how would they behave for a specific vaccine, then can we make a more personalized vaccination scheme where we give you the specific vaccine that would best augment your protection. And so maybe instead of having one vaccine that we all get every year for flu or for COVID, maybe we can have 2 or 3 options. And for each person, we tell them, you should get vaccine 1, whereas I should get vaccine 2, and your neighbor should get vaccine 3. And that way we'll increase everyone's antibody responses by giving them the specific vaccine that would be best for them. And finally, my favorite part of these methods is that we can go in and we can see what exactly did we learn at the end of the day. Because all of the tools that we use are highly interpretable. So it's not just some opaque box that you can't peer inside. You can actually ask, what are the rules? Who responds the best to a vaccine and who responds poorly? And can we take that in order to design the next generation of vaccines so that there are no holes in the population's immunity, so that everyone is covered by the vaccines that we provide and they get the best, strongest, most durable response that we can create for them. And so going back full circle, what we're doing is we're taking the many studies that we've done in the past decades, in last year, in this year, and that we'll keep on doing next year, and we're going to use those studies in order to predict future responses. And I think as a physicist, there's a really nice symmetry here. Even though I didn't show you the actual equations that we're using under the hood, there's something beautiful when you use the past to predict the future. And it makes this kind of time symmetry that's very powerful and becomes very effective. And so with that, I'd like to thank you all for your time, and I'm happy to answer any questions.